Everyone, my name is Desiree Warner. It is January 13th, 2019, and my assignment today is to talk to you about what I went through this week, and it's pretty much a reflection and a review of the exercise that I've completed. This week pretty much talked about experiments and safety and cultures and the environment. It also gave us a bit of a microbiology terms, calculating time allocations, and again, incubation spaces. I have noticed first of all, the calendar, I think is going to be very helpful for me because it gives me a bird's eye view of what's expected of me, gives me a mapping. It helps me to map out my time, actually, mapping out the next 8 weeks. What I'm finding is that for weeks 2, 3, and 4, mainly because it's going to be a lot of work, those 3 weeks are going to be very intense because we're going to have 2 different cultures being incubated at the same time, 2 different experiments running. So it's going to be very pertinent that we stay on top of this work. Putting in the due dates works for me because it really keeps me on my toes as far as when deadlines are approaching. Don't want to wait till last minute, certainly not. The pouring times, because I'm a visual learner, I see it and I get it. I can grasp the concept of what needs to be done. Location, it's very important. Doing the experiment allowed me to really examine the areas in my house as far as temperature, draft, and just safety, as far as children and pets. I have a cat, I have 2 toddlers, I have a 4-year-old, I have a 3-year-old. They are very curious, so I really don't want them, or they should not be venturing into incubation, petri dishes and stuff. So my experiment, it needs to be away from them, it needs to be safe, they cannot be near them. I explored my bathroom, I explored my bedroom, I explored the kitchen counter, which would be, I have a large space, but it still grants them access. Then there's the cat who can climb and jump, and she does that. So I don't want her to get into my experiments and start spelling, cross-contaminating, and then it's a mess. So I have to be cognizant as to where I place my stuff. Also, temperature, it's very key. I cannot place anything below 20 degrees Celsius. It will not be efficient for the microbes as far as maximum efficiency for the growth of the cultures. Draft, too much high wind will dry out the cultures, and direct contact, direct light will not, it wouldn't maximize the conditions needed for the growth of the culture. So I need to protect myself. Back to that. So not just the environment, I need to protect myself. I need to wear gloves. I need to wash. I need to wash the area before I start working. 10% bleach solution. I need to wash my hands and my forearms, even with wearing gloves, thoroughly with warm water and soap. I need to properly label dishes, label the bottom of the jars, the agar jars, and not the top of it, but the bottom. When I'm washing after, wash them with the bleach. If I'm going to be using these equipment, reusing them, soak it in 10% bleach, and then rinsing them in tap water, and then securing them, wiping down the area. Did I forget anything? Let me look at my notes here. If I spill anything, I am to cover, saturate paper towel with the 10% bleach solution and cover it and leave it there for 15 minutes before wiping it up and wrapping it into a plastic bag and discarding it in the garbage. During my experiments, of course, my children will be kept away, and you know, hopefully my mom can help me watch the kids while I do the experiment. Yes, we talked about labeling. I'm just looking at my notes here that I made prior to recording, and I believe I covered everything. You know, the point of the safety is to prevent myself from getting sick and to prevent cross-contamination of the cultures, because you really want to procure accurate results if I'm going to be dealing with cultures. That's it. I want to maintain a clear space, because if I work in a space that has clutter, it's just a disaster, a recipe for disaster, you know, causing spills. But so a clear, clean environment will lower the chances of any accidents or spills occurring. And I believe that's it. Did I miss anything? I don't think so. Thank you for your time. And I just, just a last thought. I just think that proper planning would be key to this, time-wise and space-wise. Those are the two variables. Your time and your space would be very much key to successful experimentation throughout this next 8 weeks. I think I covered everything. Again, my name is Desiree Warner, and thank you for watching.